Hello, and welcome to the Scientist.com webinar, Overcoming Challenges in Biobanking Part 3, Working with Industry, presented in collaboration with the UK CRC Tissue Directory and Coordination Centre. This is the third and final webinar in our series, and our previous webinars can be accessed via the UK CRC and Scientist.com blogs. Today, I'm going to give a brief introduction, followed by presentations by our fantastic industry panel on the relationship between biobanks and industry. Our panel consists of Dr. Kirstin Goldring from AstraZeneca, Marcela Aguilar Matta and Paul Schröder from Novartis, and Jessica May Sims from the UK CRC. After the presentations, we'll have a question and answer section to give you the opportunity to engage directly with the panel. So please feel free to type your questions into the Q&A box at the bottom of your screen as you think of them, and we'll be sure to answer as many as possible. Those of you joining us for the first time in this series, my name's Matt McLaughlin and I'm the VP of Compliance and Categories at Scientist.com. I've been involved in drug research and discovery for the past 20 years. Prior to my role at Scientist, I was a scientist and senior scientist at AstraZeneca for 12 years within the R&D genetics and personalized healthcare biomarker departments. Within my role in Scientist.com, I'm responsible for the creation of our award-winning Comply platform that supports most of the world's large pharmaceutical companies access human biological samples from commercial and academic biobanks. I also lead a team of scientific experts who support researchers on a daily basis to identify and source the services they need to drive their experiments forward. Just to give you a brief introduction as to Scientist.com, if you're not aware, we're a software solution that's designed to support scientific research and was founded in 2007. We're headquartered in the San Diego area, and we have offices all over the world, including the UK and Japan. We currently have approximately 60 full-time employees, supporting over 80 biopharmaceutical companies, including 24 of the top 30 pharmas. And we provide access to over 3,300 different suppliers in more than 4,000 research areas. I have the great privilege today of introducing our speakers. So our first speaker today is Dr. Kirstin Goldring, the Director of Human Biological Samples, Compliance and Governance at AstraZeneca. Kirstin joined AstraZeneca 6 years ago in the role of Principal Scientist, Human Biological Sample Governance and Strategy. Kirstin now leads the AstraZeneca HBS Compliance and Governance team, supporting the use of HBS across the business. She works with the biobanks globally to ensure that AstraZeneca scientists can access the samples and data that they need in a compliant way. Kirsten has worked in the biobanking field for over 18 years in different roles and sectors. Following Kirstin's presentation, we'll have two individuals from Novartis. First will be Marcela Aguilar Matta, and then followed by Paul Schröder from Novartis. Marcela is a global sourcing manager focused on supporting research at Novartis. Her role includes the human tissue category, and she has more than 15 years experience across several industries, including pharma, agriculture, and consumer products. Paul works with HBS suppliers globally to ensure Novartis researchers have access to tissue samples and data that meet the highest ethical principles as set forth by the Novartis Human Tissue Supplier Code. Paul is part of the Novartis Human Tissue Council and has previously worked in different compliance areas within pharma and academia. Paul is a biologist and biochemist and holds a PhD in applied medicine and in bioinformatics. And to finish things off, we'll hear from Jessica May Sims, who is the Engagement Manager for Collaborations at the UK CRC Tissue Directory and Coordination Centre, also known as the UK CRC Tdcc. Tdcc strives to maximise the use and value and impact of the UK's human sample resources by ensuring their visibility to the research community. There, Jessica works with external stakeholders to join up policy across the UK's biomedical research landscape to achieve this goal. She brings a background in policy research and patient and public engagement to the Tdcc's multidisciplinary team. With that, we're excited to jump into these presentations and hear from these qualified panelists. So now I'll turn over to Kirstin for her presentation.
Thanks, Matt, and thanks for inviting me to be part of this webinar today. As Matt just said, I'm the Director of Compliance and Governance at AstraZeneca, and I will give you a little bit of context about that role and where it fits in the business in a second. But just also to highlight that previously I have had roles in biobanking and academia as well. Okay, so I wanted, as I said, to give you a little bit of context into how we use human biological samples at AstraZeneca and how we support the use of them across the business. We actually have 3 groups that form our biosamples department, which is part of precision medicine and precision medicine and biosamples at AstraZeneca, and we're responsible for HBS activity and oversight. I do head the governance and compliance team, as you've already heard, and we're responsible for our HBS policy positions and governance, supporting the due diligence processes and compliance tools around the work that we do with human biological samples. We also closely work with our colleagues in clinical sampling and alliances, and they're there to support the study teams on our own clinical trials where samples are being collected to cover all these the sampling aspects as part of our ongoing clinical studies. The samples then come into our biobanks, and they're the 3rd group within biosamples, and we also work very closely with them. They're responsible for the operational aspect of all samples coming into AstraZeneca, and I'll talk a bit more about what those samples are and where they come from in a second. But we have over 4 million samples currently in our biobanks globally across AstraZeneca, and these cover all of our therapy areas. So really, human biological samples are a very key part of the full end-to-end R&D process for discovery and development of new treatments and diagnostics. And over the past few years, there's been increasing demands for samples, increasing requirements to be adaptive as clinical trials are changing as they're developed, and there's a real need for samples and to use them as part of the studies, but also in that development process. So we're working very hard to ensure that the scientists constantly get the samples they need for across the business to cover that full end-to-end piece. So how do we access these samples and what samples do we use? Well, as I've already mentioned, we obviously run our own clinical studies, and from those we collect a number of samples, both for use on study and future use. But we also access a number of samples externally. We have a lot of research collaborations, and we're sort of reviewing around 30 to 40 research collaborations a month that have some level of HBS involvement in them. And our role in that is to ensure that any samples used as part of these collaborations are done in line with our policies and standards around human biological samples. Some of those samples will come into AstraZeneca, other samples will be used by the collaborators, and our role there is just in interest and obviously in the results of that. And sometimes we arrange for analysis to be carried out elsewhere. So we also, as well as the research collaboration, source material on a fee-for-service basis. And that's obviously where the AIM platform comes in, or the scientist.com platform, through which we access all of our samples on a fee-for-service basis. And these come through commercial suppliers and obviously hospital and academic biobanks. So the biosamples group sit at the heart of all that work around human biological samples, whichever route they've come into the business. But really the role of my team is to support the due diligence processes around those samples and how they come in. So I wanted to spend a little bit of time just talking through how we do that. So of course there's a requirement initially to access or use a particular sample, and we need to identify where that sample will come from. And when we then need to run through the due diligence process, be that a collaboration or materials that we're sourcing. And then we have an approval decision to that, and obviously the relevant contracts that then go alongside those samples and bringing them into AstraZeneca. As part of that due diligence process, we look at some key aspects which I've highlighted here. One, of course, is the consent. And from our perspective, the important thing about the consent is that it covers the fact that samples may be shared with or used by commercial companies such as AstraZeneca. And that's really important to us because we're keen that anybody that's donating samples is clear that they may end up being used by industry. And that's partly around reputational risk, but also, of course, making sure that the patients are aware, and if they have any concerns with that, they wouldn't, they'd have the option not to give samples under those circumstances. There's also some requirements in the consent depending on what we might be using the material for as well. So there are specific ethically sensitive areas where we would want to see language in the consent. For example, if we were using them for genetic analysis, we want that covered. If samples might be used in animals, that's another key point as well that we will look at in the consent, as well as other areas. We also look at the legal, regulatory and ethics requirements to ensure that appropriate ethics was in place for the collection of the original material, but also that the appropriate ethics is there for the use of the material for the purposes for which we will bring them in. And we have global policies and standards around the use of the materials and the requirements around these due diligence processes to meet these standards. Of course, then we have the contracting arrangements, and so through the scientist.com platform, when we're sourcing materials on a fee for service basis, that is a standardized contract. And we also have similar contracts when we're working with collaborations as well. So I just wanted to quickly take the opportunity to highlight some other considerations when working with industry, and obviously the things that biobanks may have some concerns or question about when sharing samples with industry. We've run through the piece around informed consent and why that's important to us. Obviously there is a reputational risk component to that. There's also speed of access. So I think some of the things that is important for us to do is identify potential sources of samples and have contracts already in place, and to have reviewed those documents ahead of time, because I think that's really valuable to both parties, in that certainly sometimes when we have requests for materials, we need to access them fairly quickly, and projects change as well. So it's important that those things are set up, and it enables us obviously to quickly access samples, but it also means that if those pieces are in place already, then it reduces the risks of us going down a route of trying to access samples for someone that ultimately then doesn't end up happening because priorities change. The other piece, I think, is around appropriate acknowledgement, and we're working more and more internally to ensure that we are acknowledging sources of samples in any publications that have used human biological samples. And I think that's a really important point, because from a patient participant perspective, it's important to know how samples are used by industry and the value that has then in developing new treatments. Also, I think when we're thinking about collecting samples, it's important that they also have a choice about what those samples might be used for, and that they may be used with industry, because ultimately it's unethical to take and store samples because you haven't asked the right questions, and then we can't use them for certain purposes. So it's really important to make that consent informed and give people the opportunity to do these things if that is something that they are comfortable with. And also, just to highlight, I've talked a lot about collaborations, but the collaborations that we have, obviously we have samples coming into AstraZeneca, as I mentioned, and use of samples collected by third parties as part of that collaboration. But we also have a number of collaborations where we've shared our own samples that we have within our biobanks externally as well. Particularly in the genomic space, we've been involved in a number of collaborations over recent years where our samples we've collected have been used as part of direct collaborations and also part of consortiums as well. So I think that's it from me, and I'll hand over to the next speaker.
Thanks, Kirsten. Hello, everyone. My name is Paul. Thanks for the invitation to Scientists at Home. Thanks for joining today. Together with Marcela, Aguilar, Matta, we will give you a quick high-level overview on Novartis standards when sourcing human biological material for biomedical research. The acquisition, storage, the use and the disposal of human biological materials and samples are essential to biomedical research. Novartis is committed to setting and maintaining a high level of ethical, quality, and compliance standards. Let us give you a quick overview. I'm going to quickly talk about ethics and law. So, human biological materials supplied to Novartis must be collected, obtained and produced in full compliance with general international ethical principles, national laws and regulations. Providing information to the donor and obtaining the donor's informed consent for research are subject to international as well as national ethical and legal requirements. In addition, a competent and recognized ethics committee or institutional review board would typically approve informed consent documents and associated processes that suppliers use for obtaining a donor's consent and human biological material. So coming to the standards, Novartis recognizes donors and patients or their legal representatives' fundamental rights to exercise control over the use of their samples for research, which is achieved by voluntarily giving their informed consent. A model informed consent should typically contain language about explaining the research purpose, voluntariness, risk associated with the donation, usage by third parties like commercial parties, transfer to other countries, and of course, confidentiality protecting donors' identity. Informed consent should be documented in writing with the signature of the donor, legal representative, or next of kin. So how does the process work for obtaining human biological samples? For scientists.com, in a so-called onboarding process, Novartis will review and assess suppliers based on a human biological samples and short HBS questionnaire. This is completed by the suppliers when signing the HBS agreement in scientists.com. Then once onboarded, suppliers will be part of Novartis approved HBS supplier pool and visible to Novartis research community. It is then up to the researchers to select a specific statement of work, which will finally undergo a second assessment specific to the human biologic material offered before being approved and released for PO generation. This process ensures HBS being provided to Novartis passed all HBS compliance related steps and requirements and is in full compliance with general international ethical principles, national laws and regulations. And with this, I'll hand over to Marcela.
Yes, hello everybody, and thank you for this space. I will give a more procurement perspective of how we drive the sourcing of the human tissue through scientists.com. Scientists.com has been established as a preferred sourcing channel, and I want to go through some of the key reasons of why we have established this as the preferred sourcing channel. First of all, the legal framework that has been established through or via the platform has been validated by Novartis legal team. This is very important because we have validated that the terms and conditions that are through the agreements that we have established through scientists.com are the same ones that we used on our agreements. And we believe this benefits the biobanks because it's valid not only for Novartis, but for several clients. So we also see that benefit. The other important thing is the requests that go through approved vendors. The requests that are put through, it will only go to approved vendors. So we are very conscious and responsible of where we sourced the material from. And so by this, we have the right due diligence through scientists.com. And we also appreciate that the RFI that is being brought through the platform has also visibility to other pharma clients. So we try to see the benefit into the institutions that will be writing the RFI, that it would be simplified, that it would be for Novartis and other clients. Also, the compliance overview is embedded through the system. So when a researcher requests for a sample, automatically our human tissue network has this visibility, and we assure that it's well registered into our system and that we have the right tracking. And the last one, it is a centralized information system that we do keep the track on the requests, who has approved them, who has reviewed them. So this really helps us, let's say, have this good compliance on the samples that we are sourcing from. Let's say we realize the benefit of both sides of Novartis and also the biobanks using scientists.com, and that's why we encourage it to be the preferred sourcing channel. With this, I will pass it on to the other panelist, Jessica.
Thank you, Marcela. Hi, everyone. My name is Jessica, and today I'm speaking on behalf of the UKCRC Tissue Directory and Coordination Center, or as Matt already said, Tdcc, make it a bit easier. So our remit is to improve biobanking in the UK. So our flagship project is the Tissue Directory, which many of you hopefully have seen. It's the UK's National Register for Samples and Data. The directory itself is driven on the data available about collections rather than a catalog of individual samples that are available. So far, we've got about 200 registered sample resources from the UK, across the UK. The collections vary. So today I'll talk about biobanks, but we've got cohort studies and clinical trials. Also research tissue banks, which must register their collections on the directory as a condition of their ethics. The reason behind that being is that if you are collecting, you can be found and discovered to other researchers for sharing. We're always seeking to improve the directory based on the researcher needs and Biobank's capability. So trying to find that balance between what could be and how things actually do work in practice. Finally, we do this work on behalf of UK Biobanks in and outside of the UK. So as the UK node of BBMRI ERIC, the European Collaboration for Biobanking. I'll be dealing with some of our work to support the access of samples. I'd like to emphasize that this means access for all researchers, whether they come from academia or industry. We'd like to support research in the UK, regardless of the organization undertaking it, as long as some of the conditions are being met with respect to patient and societal benefit. Often we hear from researchers who are not having any luck finding samples. They may contact biobanks at other institutions, either through a straightforward email, or maybe something more visually designed, a fancy PDF document proposal, and they hear nothing back. So it's like the request is entering into this black hole. On the other side, we have a situation where I'd say the majority of academic biobanks were not set up to work with industry from their inception. To them, the process seems complicated and daunting. So for example, after finally agreeing to samples that can be delivered, they get into the contract phase. It can take a really long time, both because of how things work within their host institution, and also because of the contracts being received not necessarily being fit for purpose. And sometimes these arrangements then fall through. Those situations are barriers for these partnerships to happen in the first place. So our role is how we can improve both the visibility and access of sample collections. Within the tissue directory, the information we ask of biobanks is designed to give the researcher an idea of what they're likely to find if they contact the biobank. So for example, the details of the cohort, the disease areas covered, the access conditions, and so on, to the estimate of annual access requests of the biobank. The reason we encourage biobanks to complete as much profile and collections information as possible is because with that information, a researcher can make informed contact. And I'll explain why that is important in a minute. But first, the idea is that the two sides can talk to each other. And so on this pop-up on the screen, contacting resources, you can see that the person that is in need of samples has a few different options. They could contact the biobank directly, or by using third parties like scientist.com, which can facilitate the conversation and also connect to a procurement process. From research we've carried out with researchers who use human biological samples, we know that they prefer working with local biobanks because there's that feeling that they can turn up and speak to someone about their research rather than sending a message into that black hole. Unfortunately, that's not always possible because of what someone might need isn't available locally. Perhaps if the request was approached in a conversation, rather than a list of this is what I need and how I need it, there'd be more success to the requests that are sent out. And this is because we can't assume that the terminology we use is universally understood. When we discuss desired outcomes, we can open up what is required, meaning that the requirements may be a bit more flexible than what we have thought of initially in our research proposal. Or put another way, the researcher can learn a lot from the experience of the biobanks who've seen other research projects and research design. And this discussion between the two parties allows for better context. Basically, if what is being asked for is realistic, and this might mean total caseload that you get in a month or a year. People at biobanks would know, yes, this is possible, which may not match with the research that had been designed and that they've been approached with. Obviously, from the perspective of Tdcc, we want organizations working together to produce high quality research, which will bring benefits to patients and society more generally. This hasn't always been possible because of practical reasons, such as sample availability, but also because the conditions around the procurement process. One of the reasons we set out the principles of transparency, as written on this slide, is because biobanks are not always keen to work in partnerships where the end user is unknown. And in the past, there has been the belief that going through organizations that mediate the relationships and conversations would mean the process would be a secret, that these end users would not be subject to the usual oversight or expectations placed on other researchers that might apply for access directly. In the reality of academic biobanking, when it's necessary to show value to the research process, how would a biobank be able to trace back their contribution to the research in this case? So we set out these principles to address these concerns, and we're pleased that scientist.com has signed up to them. The transparency principles recognize that biobanks are seeking incentives other than covering their costs to enter into research partnerships. For example, we have very active biobanks, which are more than busy fulfilling for projects within their own institution. And then there are the ones that may want to share or have the ability to share more widely, but are limited by their lack of resources. So staff, IT infrastructure, and experience with working with industry, the list goes on. They want to know whether the samples or data were fit for purpose. They want to know what can be acknowledged or cited. They want to be able to tell the people donating their samples, so the patients or the people coming through, that they are being used. Also, going through the hoops to provide for industry can pay off through potential future collaborations because the access has a precedent. And so it's great to have events such as this when we hear from industry partners about what they are looking for, what are their expectations, to provide some kind of context for future research partnerships. We've heard that industry is interested in working with academic biobanks because of the collections that exist or proximity to potential new donors, the linked data, and the ethical and legal frameworks which this activity is already being carried out under. Partnerships and collaborations can be mutually beneficial, and so that's why we encourage the two sides to meet and discuss to see what potential there is for producing high quality research. Over to you, Matt.
Thank you very much, Jessica, and thank you to all of the speakers today. With that, that brings an end to the presentation section of the webinar, but we've been receiving questions throughout. So we're going to move into the Q&A section now. So please, if you do have additional questions, feel free to submit them first through the Q&A functionality in the webinar portal. So to start with, a relatively simple one, which I will direct to Kirsten, is does industry always prefer to source samples from commercial suppliers?
Yeah, thanks, Matt. No, so certainly, obviously, we do source material from commercial suppliers, but we're also very keen, and I guess it's linked to some of the transparency pieces that Jessica alluded to. It's actually much easier sometimes to have those direct relationships with the source of the material as well. It helps some of those conversations about understanding exactly what's required, and making sure that obviously there's an understanding of where samples are coming from. So we do have quite another number of interactions with both hospital and academic biobanks, and obviously it's important that we try to do that where we can directly.
Can I just for the sake of completeness, Novartis, would you say that you would agree with that, or is there anything you would like to add?
I think Kirsten really covered that very well. So I would really echo what she said and what also Jessica mentioned today.
Fantastic. So one of a follow-up question. This gets. We shall see how the answer goes with this one. So thank you to Sharon for submitting the question. I'll open this up to the floor. So any of you jump in once I've finished. Has using a platform like scientist.com increased your access to tissue, and has this increased the speed of accessing it rather than not using it?
Yeah. I mean, I'm happy to pick that one up. Yeah, absolutely. It has very much increased the speed, because I think I alluded to in my presentation, obviously the more you can have the information available ahead of time, and to have done all these reviews and to have the contracts in place, it makes those interactions much easier and much quicker, and it gives you obviously we can have a wide variety of potential suppliers available to go to for the different types of materials that we might need in different scenarios. So definitely yes, and we do mandate that all of our sourcing is through the platform as well, because obviously we need to do these pieces anyway. We need to collect the information, we need to review the consents, and so that's all required anyway. It just streamlines that process and makes it much more efficient.
Fantastic. And actually, this leads me on to another question that was submitted, and it's in a similar. Sorry, it's asking a related question. But is all HBS sourced through scientist.com? If not, why? And what is the supplier qualification process for the vendors that aren't accessed through the platform?
So within Novartis, scientist.com is the preferred sourcing channel, and we do try to drive everything through scientist.com. Just because, as the last question, we find a lot of efficiencies on time, and it really makes it a lot easier on the process. We do have exceptions, but it's managed as an exception, and the approval process is a lot more complex, and we do need to get a higher approval. The RFI or the questions would be similar, the terms and conditions would be similar, but the recollection of the information is more complex. So we would not recommend, when it's not a collaboration, to go outside of scientist.com.
Yeah, it's very similar for AstraZeneca as well. As I said, apart from when we have collaborations, because then again, the things we would look at and the reviews we would do and some of the contract clauses would be similar, but of course on the more collaborative piece there are other aspects too. So it's the fee for service pieces that are all mandated through the platform. But yeah, collaborations, they do happen slightly differently, but actually the processes and principles behind them are very similar.
Jessica, just to give you a heads up, I'm going to direct this one at you. We had a question in, can US HBS suppliers register with the Tdcc?
So as far as we're concerned, we're interested in sample collections that are located in the UK. And so if there are entities that are operating in the UK and they're storing samples locally and they are subject to ethics, such as being registered as a research tissue bank, then yes. If the samples are not registered or kept locally, as in domestically within the UK, then no.
Thank you. Hopefully that answers the question in full. So to change tact a little bit, this will be directed probably at the industry folk. Is there a difference in having ethical approval for commercial research and providing tissue for third-party use? Is wording within the consent form crucial here, and if so, what could be better wording than generically saying working for or use by commercial? I guess the key there is sort of what are your feelings with regards to the best way to communicate that to the donor?
I can take that one, Matt. Thanks for asking. It's a great question. So I've come along with a lot of consent forms and reading through them. So they're very, very diverse. So sometimes commercial research is mentioned. Sometimes it's mentioned about usage of third parties. So what we definitely look at is that commercial research is permitted, and it's something that is usually written within the consent form or the PIS. So it really depends, and it's, as I said, it's very, very broad, but it should definitely be covered. Yes.
Thank you, Paul. This question's actually been submitted specifically for you, Kirsten. So I'm prepping you in advance. Do AstraZeneca register all incoming HBS samples that have passed compliance? Are they linked to compliance-related information, for example, the informed consent templates?
Yeah, so all of our samples that come into AstraZeneca, apart from where there's specific requirements to get them directly to the scientist rapidly because they're fresh material or something like that, come in through our biobanks. So we have an agreed process that all material comes through our biobanks, and then that links through to our approval orders through the platform. So that's captured within the biobank notification form when the samples come in. So that is linked up. Yes, for fresh samples, similarly, as I say, they might need to go direct, but we make sure that again there's an understanding of that process and that's linked through as well.
Thank you very much. The next question that we have is from Stephen Dodd. So Stephen, thank you for this. How well does having all the contracts in place before samples being taken work? Do you often miss samples as patients aren't identified because the MTA contracts in place are set up then forgotten about?
So in our part, we do need to have a contract in place before taking a sample. We would not receive any sample without an MTA or an agreement. And do we have to miss samples if patients aren't identified because of the MTA contracts in place? I do not quite understand the last part. But we do keep a lot of track of the MTA. So if the MTA it's expired, we would not receive that sample, and that also helps a lot driving it through scientist.com, because as the legal framework is set up, we cannot do a purchase order or we cannot do anything until the legal framework is in place. So we do not forget about the MTAs.
Yeah, and I think just to add this from the scientist.com perspective, when we started work in this area a few years ago, my experience previously working in industry is that contracts can be set up and they are really difficult to track. There are potentially situations where researchers aren't aware a contract exists, so they look for alternative sources, etc. We tried to resolve that challenge because through the platform, it's very clear to the researchers which suppliers, be them academic or be them commercial, do have a valid contract in place. They are then aligned with the approvals and the approved supply bases of each individual client, so that it can really facilitate a researcher understanding who they can access and what samples are available, and communicate directly to that biobank about the request, so that they can then enable the biobank or the commercial provider to submit a statement of work, which then forms the contract between the organizations. And Stephen, I've just seen that you've added some, for example, our patients identified late and it's not realized that they might be eligible. So I will hand this back to the panel to see who would like to take on the piece, but just to summarize it, the question was, do you often miss samples as patients aren't identified because the MTA contracts are in place but are set up and then forgotten about? For example, a patient's identified late and it's not realized that they might be eligible. I don't know if any of the panelists would be comfortable in following on from that answer.
Well, I'm happy to. I guess I would say that, you know, obviously because we have this link through the platform now, we know when we've got the contracts and we have that link. So hopefully that wouldn't happen now. We also, even for our collaborations, obviously have a link to our agreements that we have in place with those. And there's usually regular contact between our scientists and the sources to, you know, if there is interest in accessing material, to obviously make sure that there's a conversation if material is becoming available.
Okay, thank you. One question in the Q&A that's come through that I know is handled quite often, so it would be good to get an answer for the attendees, is from your individual perspectives, can you explain how waiver of consent samples are handled, and when they can be used, and what your concerns are with the samples with a waiver of consent?
Novartis generally does not accept waivers of consent, even if these are issued by independent institutional review boards or ethics committee. But there are certain exceptional cases when we have very, very rare samples where we would explicitly do some special review for these type of samples.
Yeah, just to say, our position is very much aligned with that as well. So I think that's a fairly common industry position.
Yeah, and just to add, I know we work with a number of industry partners, and they a large majority of them do take a similar standard to that as well. We have a question from Alison at the Wales Cancer Bank. Bear with me, this is going to be a long one. So how do we balance the biobank's need to review applications for samples and the information required to do that with industry's concern over confidentiality, if that is the reason that pharma doesn't give sufficient detail in applications? And she goes on to say, our biobank is very willing and open to work with industry, but have an obligation, as per her RTB ethics approval, to review all applications for samples to ensure good use of the bioresource. The one issue that they find is with industry is that they are unable or unwilling to provide the sufficient detail of how they will use the samples to allow a review. how, from the industry perspective, how do you sort of encourage your researchers to overcome that problem to enable access to the samples?
Hi, it's Kirstin. I mean, I think that's a really good question, Alison, and certainly I think what we would do, and so from our perspective, if there is that issue, there is obviously sometimes things that we can't share, but I think usually we should be able to work with our scientists to understand what we can share to be able to give you enough information to do the appropriate reviews that you need to do as well. So I guess what I would say in those scenarios is, make sure you kind of reach out to the centralized teams who can support that dialogue between the scientist and the biobank to work out exactly what you need to know and how much we can share appropriately.
Yeah, I can pretty much echo that. So we have a specialized team in Novartis which basically deals with these types of interactions, and they usually it's a pretty good process and that works between biobanks and the researchers.
Thank you, guys. Another question that's come in. How do you keep track of permitted sample use as per the donor consent? So how do you make sure that when you source a sample that any restrictions, etc., are communicated through to the end user?
I can take that as well. So on Novartis, we have a standardized process. So each of the human samples that comes in is basically reviewed in terms of informed consent, IRB, and we really check each of the restrictions that are written in the informed consent. And all these are basically translated into a system. And what we have the possibility to track the samples down, we always are in position to react to whatever restriction applies to the informed consent. So yeah, I would say the answer is yes, we can do that.
Yeah, similarly for us, so when we do the reviews of the individual orders now within our compliance review, we capture any restrictions, for example, genetic use in animals, derivation of cell lines within the approval for that particular order, and then so when the information goes through to the biobank, those restrictions will be linked to those samples that came in under that particular order as well.
And again, just to add from our side, we because this is an absolutely critical requirement in the use of samples, part of the process through submitting a statement of work in the process at scientist.com, there is a field that enables you to communicate extremely clearly if there are any restrictions on use to the end researcher, so that they're fully aware of what their permitted use is and can make sure that it aligns with what they're intending to do as well. And as part of that discussion, we had a question coming in that's linked to it. Again, I will open it up to the floor, but has using scientist.com improved and or streamlined that tracking process for you?
In our case, it has, and then it has also given us visibility throughout across Novartis. So we have different business units, and we try to use this across all business unit, and it has standardized the process, so it gives us visibility, and we can track all of the samples that come in through this different business unit. So it really has helped to standardize that process.
Thank you. Had a question in from the attendees as well. What is the ethical governance framework around the research work in the industry, such as application through ethics committee? Would anybody, I'll open it up to you, would anybody happy to take that one?
Yeah, I'm happy to take that. I mean, particularly in the UK, we have AstraZeneca, as obviously we've mentioned, we have our own internal biobanks and samples come through those. So we have a mechanism in the UK. We have a research tissue bank approval, which we report annually the different projects that we use material on back through to that committee. But of course, it's dependent on the country in which the biobank is based as the mechanism we do that. But similarly in Sweden, we have some project-specific approvals for work that we do in Sweden. So it will depend on the country, but we do have mechanisms for doing that.
Thank you, Kirsten. I know we've done quite a lot of questions, and I'm going to do another couple before we wrap up. Kirsten, one's been submitted for you specifically related to your presentation. Would you be in a position to share roughly what percentage of the 4 million samples are accessed annually?
Oh, that's a really good question. And quite a difficult one to answer. What I can say, we did actually look at this. I gave a presentation, I think, at European Biobanking Week, probably last year, maybe the year before, on this. And actually, we looked at this, but actually, it sort of depends largely on the samples. So, the samples that we've got coming in from our clinical trials, some of them are coming in because we've already got planned analysis, right? So those, obviously, it's relatively high because we know we're bringing those in for particular analysis anyway. Also, for some of our genomic samples, we're collecting them with plans in place. So I think something like 50% of our genomic samples that come in have been already utilised. And but obviously, some of the rarer collections we bring in and some of the other pieces, they are less, depending on the sample types. Where we've got lots of biofluids, we would, obviously use a small percentage of them potentially, and some are deliberately brought in to look at a later time point. So it will depend really on the sample collections, and I have got a presentation on that. So if someone's specifically interested in the details of how we break it up, then I can certainly share that.
Fantastic. And if you are interested in that, as you can see on the screen, Kirsten has very kindly offered up her email address. So just pointing you in that direction. Second to last question for the seminar or the webinar today is, can you provide any recommendations for commercial sample providers that can deliver ethically collected sample? I think from the recommendations that we would have is that certainly if you're is making sure that you have your processes in place that have been developed, that you have your if you're sourcing as a commercially sample provider, you'll either be collecting yourself to make available, or you'll be sourcing from a primary source, and making sure that you've done all the proper due diligence checks with that on that source, making sure that the informed consents templates are being implemented correctly, that there are appropriate ethics approvals, that they have a proper tracking system, etc. It's really going into depth into making sure that you have you don't take anything on face value. You do the due diligence checks. That's exactly the reason why the industry implemented the scientist, worked with us to implement the request for information, to go through and understand the source, understand their processes in more detail, and make sure that you're fully confident with that. There are also a lot of resources out there that can help you with that. There's the UK CRC who are brilliant at providing support to biobanks. You have your local government and regulations and legislation. So in the UK, you can reach out to the HTA. You can also reach out to some international organizations that would be able to help as well. So to wrap up, the final question, I think, is one that I've heard a lot as I've gone to conferences over the years. So this one, just to wrap up, is why does industry not always share the results of the research with biobanks? So Paul, I will hand that one to you to start.
Sure. Thanks, Matt. That's a very, very good question. So what we have experienced is that usually, I mean, the main goal when using samples or data from biobanks and for the main goal are publications, and data will be shared by means of publications. And we have a process in place that we make sure that biobanks and donors are acknowledged in the publication. So I would say that, yeah, this in these terms, we are basically like sharing the results in this sense. Does that answer the question?
Yeah, I think it gives certainly a good perspective. I think that one of the things that I've noticed, and Kirsten, I don't know if you would concur, but there's certainly been a move by industry to be more transparent. Kirsten in a presentation mentioned around making sure the acknowledgement and the that they acknowledge properly the source of the samples where possible. I think that the difficulty of sharing results from our perspective is that is ultimately the IP generated by that client, and they protect it very carefully. So Kirsten, is that something that you would concur with, or do you have a different opinion completely from what Paul and I said?
No, I don't have a different opinion at all. I think that you're right. I mean, certainly I know within AstraZeneca, you know, the number of publications that come out from AstraZeneca are quite large now. So certainly a lot of those results are sort of shared back through that mechanism. Obviously directly sharing individual results back with biobanks probably is more likely in some of the more collaborative arrangements, because obviously that's probably the purpose of these collaborations to some degree anyway. And certainly in those scenarios, I guess we would share them more directly back. But those that we're bringing in, you know, as we sort of said, for those specific research projects, would certainly largely be shared through publications as well.
Thank you. And actually, I've just seen one question that I do want to respond to, coming from Keith. So Keith asked, we currently have to upload compliance documents, IRB, ICF, for every transaction through scientist.com. Are there any plans to institute a document reservoir within the platform to house these type of documents such that the customer can choose them without individual uploads on every transaction? Keith, just to let you know, this already exists. That is the human biological sample request for information. You're able to upload as part of your request for information any relevant ICF templates, etc. And then when you're completing the statement of work and you have to answer the compliance questions as part of that, you can very simply just refer to the file that you've uploaded. So you don't have to attach them each and every time. You can provide an updated RFI, attach them all once within that. That will enable the individuals like Kirsten at AstraZeneca or Paul and Marcela at Novartis to review them independently from each other and make their own decisions based on their own individual policies and procedures. And then should you go to submit a statement of work, it's very simple. You can just reference the name of the relevant file and that saves you having to upload. So I hope that was helpful. And with that, that completes our questions for today. So we're going to go ahead and wrap up the webinar. I'd like to thank all of our presenters for sharing your knowledge and your experience and answering the questions. I'd also like to thank all of the attendees today for joining us and for submitting those questions. If you have any additional questions or would like to get in touch with us, our confirmation is available on the slide, as you can see. And I will, with that, I'll also let you know that this webinar was recorded. We're going to make it available for later listening. And thank you so much for joining the Scientist.com and UKCRC Tdcc webinar series. And we wish you all a safe and relaxing rest of your week. Thank you very much.